Fungi Maps Funginar number 3 with Dr. Jordan Bailey. Good evening, welcome Fungi Map members, visitors across Australia and overseas. We acknowledge the traditional custodians throughout this land and pay our respects to elders past, present and emerging. We also recognise the deep cultural and scientific knowledge of Australian fungi held for millennia by Australian Aboriginals, Torres Strait Islander peoples, and we welcome you all here today. My name is Susie Webster and I thank you all for joining Fungi Maps Funginar lecture series. At any time during today's event, please feel free to submit your questions by clicking the Q&A button at the bottom of your window, and our hosts, myself and Sophie Green, will raise these with our speaker during a brief interval in the middle and then again at the end. Now, our interval is a little different tonight as Jordan has to dash from the Herbarium building across a car park to the Live Cultures building, and there's more interesting things there as well. That brings us to our topic for this evening: New South Wales Plant Pathology and Mycology Herbarium, Specimen Preservation and Purpose. The work of herbaria is often never heard, dismissed and neglected by both the public and the governing bodies that fund them. But these are great repositories of extraordinary wealth of information about our environment and species. They house collections and descriptions of most of our plants, a lot of mosses, lichens, ferns, rusts, some slime molds, and a portion of our fungi. Just to give a few stats, in Brisbane we have the Queensland Herbarium, or BRI. In the Brisbane region, there are approximately 10,000 known plant species, and currently there are about 4,000 fungi collections, most with IDs from the same area. Now, as a lowball average, there should be about 4 fungal species per plant. So that means we could be short some 36,000 fungal species just for the Brisbane area. A sobering thought, is it not? So on that note, I'd like to introduce today's Funginar speaker, Dr. Jordan Bailey, Curator, Plant Pathology and Mycology Collection, New South Wales Department of Primary Industries, and based in the beautiful Orange. All yours, Jordan.
Thank you very much. Welcome everybody to the Plant Pathology and Mycology Herbarium here in Orange. So we are a government collection with New South Wales Department of Primary Industries. There's a similar collection in Brisbane and another one in Victoria as well, in Melbourne. Other than that, there's a culture collection in Western Australia, but we in New South Wales are also responsible for the collections, plant pathology collections from South Australia and Tasmania. So as you know, Susie pointed out, that funding and everything is often not secure, and these places do tend to get shut down and then absorbed into other collections, and that's happened with us. We also absorbed the forestry plant pathology collection, but that means that we end up with quite good collections here out at Orange, so silver lining. And we are part of what we're calling the biosecurity collections. So there's the plant pathology herbarium and then the insect and mite collection. So on the other side of the wall is 500,000 dead insects, but in this room here, we have over 100,000 plant diseases is what we focus on, which is fungi, bacteria, viruses. So 70 to 80% of all known plant diseases are caused by fungi. They're one of the major causes. But then we also have non-pathogenic fungi. So that's our mushroom collection, which came to us in the 60s. So the plant pathology herbarium was set up in 1890 when the Department of Ag was established. But during that time, the mycology collection, fungarium or whatever you want to call it, was housed at the herbarium with the Royal Botanic Gardens. But eventually they didn't have any more mycologists and it stopped being, looked after. And the curator from our collection at the time went there, visited to find some stuff and realized it was being eaten by beetles, and put forward the proposal that it all be moved into our collection. And so that happened throughout the 60s. For plant pathology specimens, the storage is quite easy. So we basically press and dry these diseased plant specimens, just like you would press and dry flowers. I've got a few examples here on the bench. So the greatest advantage of being able to press and dry things is that we can store them flat in packets. So you'll have a packet like this with our lovely modern looking label, or with unique identification numbers. It's identification, who ID'd it, the host that it's on, which is really, really important for us. A lot of biodiversity collections, are focused on the species, the specimen, and not necessarily the surrounding, let's say, habitat information, or what that thing was eating or growing on. Whereas for us, that's the most important data. Like, if you have a disease, you need to know what host it's infecting. If you have an insect, you need to know what it was eating, because that contributes to our pest and disease records for the state. So we are the official repository, official pest and disease record collection for New South Wales, which means that although yes, funding is, yes, funding for herbaria is not the best, but because there is a state legislated requirement that biosecurity specimens must be retained to just basically to establish area of freedom. So when our growers try to export things, other countries will ask, do you have this disease? Do you have this pathogen? And to be able to show that we don't, we do regular surveillance and surveys, both passive and active, and all of those specimens get put in a collection like this to prove that. So we're pretty good for funding, and our longevity is fairly secure, and so is my job, which is quite nice. So yeah, we'll press and dry them, keep them in packets. The advantage of packets is also that we can put things like photos there and notes, and you'll find really odd little things, postcards and whatever scrap of paper they were scribbling their measurements on. Obviously we end up with a lot of old newspaper, quite amusing sexist ads from various decades. And all of that adds that extra layer of like heritage to these collections as well. So there is that museum sort of element within them as well. Here's a bit of heritage. In the last few years, so Karen is on the camera here, she's the camera woman, and for part of her technical officer progression, she had to put forward a project, and something that she saw as a need in the collection was moving from these old cloth covered boxes to more user-friendly and what's the word? Robust plastic storage containers. So basically we had an entire factory run ordered from Sistema, not sponsoring this talk, but and got like 3,000 of these containers that are almost perfect, the same size as our original cardboard ones. So we've been moving our collection from these old boxes into these new ones. And let's come over here for a second, so you can see what this looks like. So everything's stored in packets in the boxes, and then the boxes are put on shelves. So this way, we have in this compactus some of the older boxes. So you can see it's, well, a bit of a nightmare for WHS and accessing things and being able to open them and see what's in them. But on the other side, we have just a little bit more modern looking. It's more user-friendly, especially for our pest monitoring. So I mentioned, the collection at the Botanic Gardens was being eaten by beetles. In boxes like those, you wouldn't notice unless you opened every single one. With these new plastic ones, it's very easy for us to do passive monitoring for pests in the collection. It also, that sort of modern element really helps when we are promoting our purpose to executive teams. So when we're talking to ministers and funding and research development councils, that they see this and go, yeah, wow, that looks important. They're really taking care of it, instead of, oh, what's all this old stuff? Who cares anyway? Let's just throw it out. So throwing stuff out is a problem that herbaria often face. There was a huge social media hoo-ha a couple of years ago when one of the universities was about to throw out hundreds of thousands of specimens, and I believe all of the sort of Twitter campaign or whatever forced the university to not throw it out and fund it, because there were literally collections volunteering saying, we will take it, do not get rid of it. And I think it's because they were expanding the football field or something ridiculous, So there is a tendency where executives don't understand the purpose that, they will just turf it out. So one of my greatest roles is doing things like this, the outreach, the engagement, raising our profile, and especially within DPI and within state sort of government, to ensure that they continue funding us and understanding the importance and investing in us so that we can do more with what we're taught. So I mentioned the collection was founded in 1890, but that's not our oldest specimen. Certainly most of our specimens date from around then. We got some heritage funding a few years ago to start imaging our specimens. So this is a photo here that we've taken of one of our sort of important heritage specimens. It's the oldest one in the collection, and it's from like 1802 to 1805, not so sure, because it was collected on Flinders Voyage to Coast, to chart the coast of Australia. And this is one of the other cool things about plant pathology collections and mushroom collections as well, that when you collect them, you are able to divide the specimen up. So this specimen here was a plant specimen that was collected, but when our curator went to visit Kew Gardens, where it's held, he found the florets were infected with a smut fungus, and he's able to just bring those infected florets back. Working with something like an insect collection, you have a beetle, it needs all its legs and all its antennae and everything, all its features to be identified. You can't just cut it in half and share it around the collection. So mycologists of the past, we don't do it so much anymore, but this was a really big thing in the 1900s. It's called exocardy. I don't know why, that just means to dry something down, so it's no different than just calling it a herbarium specimen. But basically, specialists in their field would create reference books of specimens. So this is the North American rusts of, so rust is rust fungi. There's a picture of one up here. So you may be familiar with these, called rust fungi, because when they infect and sporulate on the plant, it looks like the plant is rusting. And they would create these. I am not going to get through this in 20 minutes, Susie, I just realized. But we'll see. We should be all right. Yeah, so they would create these really cool, basically trading card sort of books with, they'd go out and collect all these species and then divide them all up into sets and then trade them all with each other. And, oh, you're an expert in this, and I'll get one of those books from you, and I'll give you one of my books. And they're really nice. And also, again, has that really strong sort of heritage and museum aspect, but also really important for scientific study. So something that you'll note here on it, it just says isotype. So I don't know how many of you are familiar with taxonomy, probably quite familiar, because you all tend to get into all that world, don't you? But yeah, we have type specimens here. They're probably our most important specimens. They are the species that was the specimen that was used when that species was described. So that one says isotype because they've split it up, so it's not the original holotype, it's part of. But we've got a few thousand of them, and they were a part of our imaging project as well. So they should all be up. It's on my back burner of projects, but all those images I will be sharing with the Atlas of Living Australia, so you can explore the specimens in the herbarium collection, which would be cool. We also have, as well as type specimens of plant pathogens, we do have many type specimens for native Australian mushrooms. So the mushrooms we can't press and dry, but we dry them down slowly in the oven or the dehydrator. We don't want to do that too hot because you will damage the DNA, and DNA is one of our, something that emerged as basically something that has the most potential in these collections. So, look, I can go out and chop now. Because I always say, people go, why do you need so many, or why do you need duplicates, or whatever, but people didn't know in 1890 when they started the collection that we were going to discover DNA and be able to do what we can do with these specimens. And you can see here on this chart from the purple bars that this is just Google Scholar hits for the word herbarium. And when we start being able to get DNA, successful DNA extraction from herbarium collections, the use of collections skyrockets. The other line is accessions into the collection. So people are accessing collections more and more and more. It's an instant resource. You don't have to go out collecting. Someone else has done all the hard work for you, but they're not putting back. So it's a global trend. I don't know, but people are busy or they don't value it, or people do tend to do all this collecting, shove it in a fridge, finish their project, move on to another one. So part of what I try to do here is, encourage DPI staff to submit specimens. That's what we're really pushing for, is highlighting, our importance as a repository, not just for biosecurity specimens, but our researchers' specimens as well, and securing those for, future research and evidence of their own research. The mushroom mycology collection gets really cool because you go beyond that plant pathology interaction. We do have things like the honey fungus, the armillaria, but then we get into cool little native things. So here's a fungi mat specimen, the Cyptotrama aspirata. And then other, human mushroom, human fungi interactions. So we've got the death cap there, and also a little local little psilocybe magic mushroom there. And then I just wanted to I have the little Cyptotrama out because this one's cool, because you can see when we air dry down mushrooms, they're not, their former glory is not really translated well into their dried form. But we've been playing around with freeze drying. So when you freeze dry a specimen, you basically take it from that crystal structure straight to the gas. Oh, sorry. Which means that the shape of the mushroom is preserved. So you get really good, hang on, we're not focusing very well, kind of. But the gill structure is maintained, the external structure, the cap shape, and all of those things that are really important for identification. And the color is actually retained quite nicely as well. We freeze dried that like 3 or 4 years ago, so it's still looking very nice. How are we going, Susie?
I think we're going okay. Can I ask you a couple of questions as we go, Jordan? Yeah. So we've got a question. Is it getting more difficult to make a collection, and could that be contributing to the decline of specimen deposits at herbaria?
It depends what's more difficult, because I know we have very active Sydney Fungal Study Group and the Central Coast Mycology, and I know that they're collecting, and my predecessor has a good relationship with some of those members and does, they work with him to help identify some things, and then he'll put them into the collection. I'd like to see a stronger relationship in that sense. And then we have some very good relationships with like Roy Falling. He comes and does his Bowley collecting and deposits everything with us. I would say from a mycology point of view, probably the level of research has actually dipped, because there's less of an investment in that space. They talk about golden age of botany, the pursuit of science for the sake of science, and you just go out and someone give you heaps of cash to just go collect mushrooms and name them all, and that's harder and harder to get things to do. So it's more about research with application, which is why I'm working in agriculture and ended up in biosecurity, because that's where the funding and support is. But you'd think with plant pathology, we have heaps of plant pathologists in the department. We have heaps of agronomists. They're out in the field every day. They're seeing stuff all the time. They just don't know we want it. Because a lot of them, they say, oh, you don't want that. I see it all the time. And I'm like, yeah, we don't have any specimens of it, because everyone says we see it all the time. You don't want it. So I think it's just over the last, few decades, a bit of a disconnect. They decentralized the government, moved us out to Orange, the diagnostic lab stayed in Sydney. They started charging for diagnostics. So we got less specimens through the labs. It's just been a number of things that we've got to sort of address. Yeah.
So it sounds like, as well as getting more department employees to submit specimens, it's probably general public as well that you would like to be contributing more towards your collection.
And people, I know that sometimes there is an attitude of, like I went on when I was at there was a pathology conference in Boston, and we did a foray, and we collected tables worth of stuff. The tables were absolutely covered in mushrooms from this little university park thing. And it was just this beautiful biodiversity, and we sort of half ID'd them and went through the keys and whatever, and then just put them all in the bin. And I was like, so we just spent all day collecting these and all afternoon IDing them, you know, and does the Harvard University collection not want them? And the girl was like, oh, you can't just put anything in the Harvard collection. So there is that, also that barrier where people think, you know, oh no, you know, it has to be important.
Yeah.
I'm like, well, the whole point of these collections is through time and space. If we're not constantly collecting, we end up with gaps in our knowledge.
Yeah, and who's to say what's
from a conservation point of view, you know, yeah, monitoring species distribution and diversity, and from a plant pathology point of view, monitoring the effects of climate on disease spread and all of that sort of research, that if you've got gaps in the data, yeah, you're not going to be able to do that like big data study that we're seeing happen more and more with things like GBIF, the Global Biodiversity Information Facility, and the iDigBio digitization. They're just harvesting all this data from all these collections. So we need to put more stuff in, you get more stuff out. Yeah. Absolutely.
I've got a couple more questions for you. And also I want to know what that giant fungus is right there. Yeah, what is that one?
Yeah. Well, so this was, we work with the Orange Naturalist Society, and they've been doing a survey for Mount Canobolas to stop like bike tracks being put in. So they bring me random mushrooms they find from time to time. And this is a later porous, so it's like chicken of the woods kind of group. And you can see where it was connected to the back of the tree. Oh, its back was on. And it came out really nicely. Like they brought it to us this way. And I'm guessing the tree was potentially on fire that it was attached to, because it's got all these char marks, and it seems to have actually just dried it out naturally and stopped things that were eating it. There's some little munching holes. So that's the biggest problem. You know, you get a specimen like this into the collection. If this was fresh and wet, it'd take forever to dry down, and the amount of critters that fall out of them is quite alarming.
All right, we've got a question. Does freeze drying affect the DNA, and does it affect cell structures?
So it preserves it better. I'm going to go into freeze drying a bit over in the cultures, because we actually preserve the cultures under vacuum when we freeze dry, and then revive them. So the DNA is helps. We haven't, I mean, I've not done anything looking at, look, basically when you dry it down at room temperature between newspaper, you can still get DNA out 100 years later. So drying it down in a controlled, you know, less shocking environment, like slow drying, will preserve it longer term better. It's a better system, yes. So no, it doesn't affect it. Yeah.
All right, and I guess we'll hear more about that over in the culture lab. We've also got a comment that it's great that you'll be sending images to the ALA. What about the specimen data itself? Is this available through the ALA?
Yeah, so we're working on that. Previously, being biosecurity collections, I mean, obviously the mycology stuff, yes, that's a no-brainer. But the biosecurity things, there was all this idea that it was sensitive data and it's trade sensitive and our trading partners will get a hold of it. But it's all publicly available anyway, and we're making decisions publicly available, as in if someone requests it from overseas as a trading partner, you know, we supply it. So it's all online on the Australian plant pest database, but that's a secure database with restricted access. But yes, we will be looking. We've just moved to a new database, so we're still working on cleaning up our data and reconnecting online and all this sort of stuff. But we want to do a public-facing database for ourselves, and then also share this with ALA, Virtual Herbarium, all those places, because yeah, the more people that know the stuff here, the more people use it. So since revamping our website and saying we have a culture collection, Karen's been very, very busy the last few years. We get far more requests and people putting stuff in, and industry, like private companies, requesting the services for freeze drying and preservation. And so yeah, there's no point in hiding it all away, because then nobody knows it's here to use it. So accessibility is one of our biggest core values, or whatever you want to call. So yeah, it is, and adamant that it's, you know, free access, free open access, because you know, the people that put it in here have already, it's already usually been taxpayer funds. that have like paid for the research or industry funded, and then it's in here and we maintain it as a service. But yeah, generally the international code is open data and also loaning specimens, so people can request to borrow them if they're with an affiliated registered herbarium, and we'll provide that free of charge as well. We do charge for culture sales, but that's because there's a bit more labor involved with providing them and quality assurance in the lab and all of that. Yeah.
Excellent. And finally, do you dry press and store plant material infected with, I can't even pronounce this, facultative fungi, or only with obligate fungi? Very technical question.
So, well, primarily, you mean things that like are non-pathogenic? Is that what they're saying? Like things that occur like phyloplane yeasts, or can they clarify?
All right, Tiana, can you type a clarification on that?
We do have yeast. So things like that, usually I think we would store them in culture, because that's like how I guess the submitter would have provided them. And because we're mainly, and dry cultures as well, people like, we can dry down the cultures and store them. We don't always get the plant specimen with it. When it's a plant disease, it's more important in that sense, because we want the symptoms of the host as well. But yeah, I can't actually answer that.
All right, we'll wait and hear a bit more about that.
But, yeah, I'm not sure what specimens we would have of that sort of group, if we would have the original plant material with them. Michael would be able to answer it. I should have brought him in. He's my predecessor in everything. But I'm not quite there yet with, everything that's in all these boxes. Yeah.
Also, Tiana's just clarified, if a plant is infected with pathogenic fungi, do you store the plant and the culture?
Yes. So, it's really important that we keep that host material. And it's actually one of the coolest things is you can get DNA out of the plant and the fungi. And you'd be surprised, plant pathologists aren't the best botanists. So quite often we'll have misidentified plant material as well, even with type specimens. So some of the work I was doing with the USDA, you'd have a rust specimen and they were saying it's on this host, and I was doing DNA extractions and everything, and stuff wasn't lining up, and then you go check the host and it's the wrong plant, like they've actually, misidentified the plant species. So we try to encourage, plant pathologists tend to just pick a bunch of leaves and go, good enough, but we want flowers and fruits, and now that everyone's got a digital camera, in situ photos, and as much information as you can sort of gather. So yes, the pathogen on the plant is our best thing, and then if you've isolated it from that plant, the living culture is also awesome.
Beautiful. Maybe we've got time for one more before you do your dash across the car park. As not many of the public can freeze dry specimens themselves, what is the best way to prepare and post fungi to the herbarium? Yeah,
so for your mushrooms, which is I assume what you're, well, okay, if it's plant pathogens, press dry, lots of newspaper, that's fine. That's how we do it. If you've got a dehydrator or an oven that goes low enough, sometimes you can do the oven at like 50 and then turn it off and just put it in there to help it out a bit. But otherwise, press dried at room temperature. Mushrooms, dehydrate is the best way. You can just go buy a food dehydrator and pop it in there, because it's that low and slow that we want. Anything above sort of 30, 40, you start to get into the DNA damaging. Some people do it at like 60, and that's just, we've had bad. Yeah, the DNA that you get out of it's no good, or you can't get any out because it's all denatured from the heat. So food dehydrated. So we've been working with Parks and Wildlife on the wax cap survey, and they actually express post them to us. So they go and do a survey at Mount Wilson or Lane Cove. They're only teeny, which helps. They pop them into little Chinese containers with a bit of kitchen towel, and then put them with ice bricks in a cooler bag, and Express Post just post it to us, and then I go through them all, and Karen sticks them straight in the freeze dryer the same day. So yeah, but that's because we're like in a coordinated project with yeah,
but it's good just to get those basic details about how all of us at
Yeah, and
home
you can always
can
email,
contribute.
yeah, with questions and yeah, I'm happy to answer emails about any questions about things or what to do or advice. So
Excellent.
collecting information is the most important thing.
There's a couple of questions.
when, what's it near? What trees are it near? Is it near? What substrates it on? GPS coordinates if you've got them. Yeah.
I was going to say there's a couple of questions there that I can answer while Jordan's doing the dash across the car park. So is there more you would like to do there, Jordan, where you are?
No, we can hop across. There's a couple things I might take with me, but I'll talk about them over there. That's fine.
Okay.
They overlap, so it's perfect.
Did you want to
So
talk about? Did you want to talk about
my video? Oops, sorry, Sophie.
Oh, did you want to talk about Redbubble here or at the end?
Oh, good point. No, I'll end on it. That's fine.
Okay.
But thank you for reminding me. Yeah, we'll jump over there now. I'll just turn off the video for 5 minutes. Okay?
All right, sounds good. You dash. Oh, am I on? Okay. I'll just answer a couple of the questions. So if you're collecting and you want to post them to the herbarium, there's a couple of cheats if you don't have a dryer, dehydrator. You can put them in your fungi in a paper bag and on the dashboard of your car. And that will dry out your fungal collection beautifully over a day or two. And then just pack them in a little box or a little padded bag with some cardboard around it just to keep it nice and in shape. And as Jordan said, you need to put where you found it, GPS coordinates, if there were host trees or whatever big trees were around, a species, if you can find them. You need a photograph of the environment, a photograph of the fungi, top, side, and bottom, and in its environment as well. And that should be enough information to send to the herbarium, but they also need your contact details so that they can ring you and ask you any questions if they need to know more, if they want to come and get a collection from there themselves. Because sometimes you find something brand new that nobody's ever seen before. And that gets them all very excited at the herbarium, and they start leaping around trying to get out and get more collections. There was another, citizen science. Is there a role for citizen scientists or non-scientists to play in the collection of fungi specimens? Yes, there is. I'm a citizen scientist. I do collecting, and in fact, I just found one the other day that's just been ID'd for me. It was a Calvacia, and apparently the last collection had been made in 1932. So that was very exciting. Not a new species, but a very exciting thing anyway. So you can join a local group, like in New South Wales, there's Sydney Fungal Studies Group. There's the wonderful Pam O'Sullivan at Newcastle. In New South Wales, there's what do they call it? The field gnats often have a fungi group. Doesn't matter where you are in Australia, the field gnats have a fungi group attached. There's the Queensland Mycological Society, Fungi Map have events.
There's the Adelaide Fungal Studies Group.
Yes, Genevieve Gates and her group in Tasmania. Western Australia is the field gnats, I think, that Ros, the chair for Fungi Map, is a member of and runs forays. And in Victoria, there is the field gnats of Victoria are fabulous. They've got a very strong fungal studies group. So there's lots of places you can join. And the beauty of Fungi Map is that it's for all of Australia, and we're doing our best to get representation in every state. And if you want to run an event, let us know and we'll help you out. And you can run your own event in your own state or in a country area if there's nobody around.
Absolutely.
Oh, Jordan's on the dash.
While we wait for Jordan, there was just one more question that's just come through that you might be able to answer, Susie, about don't you need to have a collecting permit in order to lodge specimens at a herbarium?
No, you do if you're collecting in national parks or conservation areas, but you can collect from your backyard, you can collect from the footpath, you can collect from your local park. The herbarium may want specific information about those areas, but my backyard, with all the rain that we've had, I had about 12 different fabulous species I'd never seen in my backyard before. Still don't know what they all were. I think there was a Limacella amongst them, which was a bit of a surprise. So there are wonderful places you can collect fungi, and we need to know what's in people's backyards. We really do. And
but collecting,
all those things are welcome.
Collecting rules might vary from state to state, though. Is that right?
They do. If you're collecting really in council areas, you should speak to council about getting a permission. Or if you're collecting on water board grounds, like around reservoirs and things, you should really speak to them about it. And national parks in New South Wales, Pam O'Sullivan might put you on her permit and let you collect under her name. And if you join different groups, they may have a collecting permit that you can become part of as well. So that covers those areas. So there's lots of different things. It's always a good idea maybe to contact the herbarium in your state first, or one of those groups, and they'll be able to tell you what the specific rules are around. And Tom Mays just commented that for permission, essentially you just need to ask permission from the landowner or the land manager, whoever that is. That's the sort of basic rule of collecting. But yeah, it looks like Jordan, I think it needs to be in writing, I think, Tom, doesn't it? And the response needs to be in writing because of a thing that was brought in 2014 and accepted in Australia is the Nagoya Protocol. So it is relevant to original custodians of land. So it pertains to ethical use and ethical collection. But yeah, Jordan's ready to go?
I think so, yeah.
All right, I'll duck out.
Equipment in this room, so there's a bit of background noise. It's okay? Yeah, good. So I've just come across on the other side of those windows is our herbarium building, and now we're in the sort of main building, but it's where most of the labs are. So this is the biosecurity containment level 5.2 lab, affectionately called the Mycology lab, even though we also do things with bacteria in here. So basically, this is where Karen lives mostly. The camera person, I'll make her be on the camera actually, I'll turn around so you can put a face to the name. And what we do in here is keep things alive and culture them. So we have the biosafety cabinet to ensure that all the cultures are pure and clean. So, basically, I don't know if anyone's ever done any culture work, but if you've got a plate of nutrient jelly, one of these petri dish, and you leave it out with the lid off for not very long at all, it'll end up looking like that because you are covered in fungi, the air is full of fungi, everything is full of fungi. So a very large part of Karen's role is when things come in, so when people send us cultures to session, she needs to make sure that they are pure. We make some cultures, plate them out, and grow them in the incubator. We mainly use, so there's a lot of different medias that you can use that have antibiotics and selective media and all of this sort of stuff to help you clean things up. But to make sure that what we're putting into the freeze dries and into our -80 is clean, we just use a general media. So everything that's there will grow. So you'll see it all is in there. But we also have this method. they call it the flap method, and basically you'll take a piece of your culture, agar, and place it underneath a disc of agar. And the idea here is to get rid of bacteria. So the bacteria will get trapped underneath, while the fungi will grow up through into this other plug on top, and then we remove that plug and put it onto a plate, and that basically cleans up any bacterial problems that we have. It can also trap mites, but they tend to scurry off and do their own thing anyway. That's why we have things raised up here, because mites are a major problem. They will climb through parafilm and into your plates, their dirty bacteria-ridden feet, and ruin all of your cultures.
Hey, Jordan, I'm just going to interrupt you for a minute. I think we're having a whole lot of sound issues. Like there seems to be sort of a bit of a feedback noise. Someone's just suggested, have you muted the outgoing sound on your laptop with the Zoom running? Would that be it? Or can you think of a way of improving the sound?
That shouldn't be a problem because it's like linked to the phone. So is it echoing or is it just noise?
We can hear you, but there's just a lot of background noise. We can just hear you over the top.
Hang on. Want me to just yell? If I just talk really loud, is it okay?
Yeah, that sounds a bit better. Just keep going, I reckon.
Yeah, I think it's just the air conditioning units and the incubators and the freezer and everything else. What should we do? But yeah, so okay. Oh, yeah, okay. So I really think, so previously I worked on rust fungi, which are obligated. They need their host, you can't culture them. So I never really did much culturing in my PhD or at Purdue or at the USDA, because it was all rust and all herbarium specimens. So here is really where I've got to experience the wonders that is culturing fungi. And you'll take something that's in the air everywhere, and it grows on a plate and just forms these most amazing colors and structures and shapes. Got this one over here. This is a photo of Epicoccum nigrum, and this is incredibly common. You will collect it if you do any sort of environmental culture. It doesn't look like that when it's in your wallpaper, but you grow it in culture, and then also you can manipulate it with different types of media and things. And there's actually a annual agar art competition where people do paintings with bacteria and fungi and all these really cool effects with different types of agar. So I said, Petri dish, basically the nutrient jelly, agar plates. There's Epicoccum in culture. These were ones that I dried down in 2019, actually, for Science Week for students. A little bit of penicillium, a little bit of fusarium. This is a pathogen, but it makes, it's going a bit brown now, but it used to be a beautiful pink color. And here's those yeasts. Smells like Vegemite, after like 3 years of sitting in that. So, with working cultures, we'll do it in the Petri dishes. But when we want to store them long term, we have 3 methods. Freeze drying. And this is what those ampules look like. So what we do is plate the fungi out onto pre-prepared plates with, it's hard to see with the glare, but basically we've got carnation leaf on there. These have been gamma sterilized, so nothing will grow on them. They are clean. So we put our culture onto there, and it then grows across the plate and finds the carnation leaf, penetrates into the leaf, which helps protect it when we freeze dry. And then that's what you're seeing in the ampules, is a little piece of leaf tissue. These are freeze-dried at a vacuum, and then it's sealed with a torch at the end. So it's double flame butane torch sealed. And Karen is very skilled at handling that. The other option is the -8 freezer, which I mentioned. This is just a commercially available biobank, microbank they're called. So it's basically these probably proprietary beads that have like little holes all over them, and the fungi and bacteria get trapped in these sort of little holes on the beads, and the special cryogenic fluid protects them during the freezing process. And then the idea is that you just snap off a little bead and place it on the plate and throw it out. So each one of the tubes has 25 beads, so we can, you know, cell culture 25 times from a tube that's in a -80 freezer like that. But not everything like those processes. So sometimes we do have to come back to the old tried and true method of slant agar storage. This is under oil, so just mineral oil. And they're kept in like a cool store, so it slows down their growth, but these are very labor intensive. So the microbanks aren't too bad. The freeze drying is a lot of work to set up. Then you stick it in a room and you don't think about it again. But you do quality assurance, but you know, you don't have to check it all the time. These is 3 to 5 years, and you're subculturing. And in some pathogens, they lose their pathogenicity when they haven't seen their host plant for, you know, iterations. And that pathogenicity is one of our main clientele. You know, they order these cultures to do like plant breeding trials. So they're breeding resistant lines of crops, and they want to check what their resistant genes are doing and all of that. They will purchase the pathogens from us to test them, you know, in the glass houses. Yeah. What else is there? Do you want to see the? Well, okay. Let's look at new things. So I was talking about the whole, we're in a really good situation at the moment with funding. Our insect hall was just upgraded. We just renovated all of this side of the collection. In that room is our molecular lab that we're just setting up. Is anybody next to?
No, I just had to peek around the corner.
Oh, look, people are still here because, Saeed's still here.
Yeah.
We're working a biosecurity emergency response at the moment, so I actually haven't done my day job since the end of February because I'm managing the Capra beetle diagnostic lab. So this horrible, well, he probably doesn't think it's horrible, but this little beetle came in shipping containers, and it's worth $1 billion to industry annually if it gets established. So we are eradicating it. In here we have our new culture collection storage room. I won't talk, very noisy. But new storage room, new cabinets, all temperature controlled, humidity controlled, all nice, and again, that modern professional looking. Do you want to do the spark testing?
Sure.
I'm going to take the camera and make Karen do the spark testing.
Oh. So part of the process with the freeze drying is to make sure that it's one thing to do all the work, but if they haven't got the vacuum out of the tubes, it's a waste of time. So there's one little trick that we do at the end of the process to just make sure that it's actually all intact and the vacuum is good. So I'm going to turn up the light and we'll go around and we'll get out the spark tester. And it works very similar to the fluorescent tube. So it'll light up with this negative vacuum, which means it's good. So we're probably in the dark.
Oh yeah, very dark.
Very dark. There's no light outside.
So we've only done this over webcam before. Oh, look at that. It's actually brighter on the screen than my eyes can see.
Oh, can you guys see that?
Yes.
So, we should go before we turn the light out. Anyway, I don't know if you can see this.
Yeah. Yeah.
See anything there?
Yeah.
So these are freeze-dried tubes, and see how they light up. That means the negative vacuum in it. Now this side here top ones lying out. Now I realise you're not going to be able to do this, guys.
Yeah.
So we know that the top one of those is no good. So, which means it'll probably be all right in storage for a very short time and will die very quickly. That'd be like, yes, that's a nice little trick that we use. So anything that's out of a vacuum, you throw away, it's not going to last.
No.
So
Yeah.
anyway, yeah.
Cool.
Back to it.
Well, just to finish, so you would have seen, like in the herbarium, there are some of these macrophotography images. We also have a lot of art. I mentioned heritage before. We have a number of artworks in the collection. You can put down there. So things that were basically in the 1960s, the department commissioned a lot of artworks for the disease bulletins and the pest bulletins. So we have entomology illustrations of pests and plant pathology illustrations, which is sort of a niche area. You know, there's not many nice artworks of plant pathogens, but there's a whole bunch of plant pathologists that have been buying them. So yeah, I've been putting them all up on Redbubble on various products. You can get socks with shot hole of, you know, apricots and smut. And also our macro photography images that we've been working on, all a part of our raising our profile image and, you know, making the website more attractive and inviting and our own brochures and marketing materials. Yeah, and it all goes back into our trust. So the collections are actually protected under a trust that was established in 1983. They have their own ABN and bank account, separate from the Department of Primary Industries, and we've been using those funds to support, not at the moment because of COVID, but internships. So giving people an opportunity to come and work in the collection with a stipend to support them. Because Orange is, you know, not very central. Yeah, and you know, we had one of those interns from, well, Lauren was here 2017, my first year, and she's still here. So she got herself a job in the entomology group side of the collection, and yeah. I think covered everything. Do we have one question?
Yes, so we did have another question. It was about whether you're sequencing fungi collected from the 1890 onwards. Like you said, your first specimen that you've got there is about 1890. Are they being sequenced?
I mean, not actively. People will request them and they'll do sequencing, like researchers who run them for taxonomy studies or to confirm identifications for biosecurity purposes. When I was working with the USDA, I was getting sequence data out of rust specimens from 1900. Rust is a bit exceptional because it has such a thick spore wall that they preserve really well and contain all that. But Jackie Edwards in the plant pathology collection in Victoria has a PhD student who's been working on sequencing from herbarium material and doing all the different extraction methods and what's the best one. And I think she's just pushed that recently, actually. So we're always improving and finding ways to get it out. And the new next generation sequencing actually works really well with herbarium material because it takes lots of little pieces and sticks it all together. And herbarium DNA is often degraded, so it's in lots of little pieces already. So, yeah.
Excellent. And something we like to ask all of our guest speakers is, what is your personal favorite fungi?
Well, I worked on rust. Just, you know, someone said, "Oh, here's a PhD. Do you want to do it?" And I was like, "Yeah, sure." But the rust fungi are just crazy. Their whole. So I really love the way plant pathogens are able to shape human behavior. We talk about, you know, exotic threats, like these things know what they're doing, but they don't care. They just see a host, they infect it. Two of my favorite stories is, well, I work with copyrights, right? And the reason the British took tea is because copyrights wiped out their coffee plantations in Sri Lanka. So, which was Ceylon at the time, which is now, you know, the tea capital, because the rust was moved with the plants from, you know, Ethiopia into there, and then the environment was good and it took off and destroyed everything. And then my other favorite one is white pine blister rust. So rusts have really complex life cycles. They will have a host where they reproduce asexually, and then a host where they reproduce sexually. And these will be two totally unrelated hosts in different families. And before DNA, it was amazing that plant pathologists were able to work this out. That, you know, it spends some time over here, and then it changes, and it goes over there, and it affects this, and does some stuff, and comes back over. They go backwards and forwards. And white pine blister rust in the US, black currants are the sexual host. So America doesn't have Ribena. I don't know what black currants are. They don't have black currant as a flavor because to save their pines, they banned growing black currants and ripped them all out. Because that was causing the population to be very genetically diverse, very, you know, virulent and exploding everywhere. So they just went, nope, no, we're banning it, rip it all out. And so you go over there and say, oh, black currant, and then what's that? Which is just bizarre. So yeah, they're really cool. Look up the juniper, sea apple rust. It's freaking weird. If you want to think, you know, you're aliens, this is one of them. It creates this nasty gall on the juniper, and then it spores out as like tentacles, and it's just the most bizarre, amazing thing to look at. Yeah.
Nice. Now, I just noticed we've got a question going back a little way. Is there a good staffed library attached to the service in Orange, and do you have room and space to grow?
Got a collection? Yes, lots. Because my predecessor, when they moved here in the 90s, made sure of that. And I have ordered 3,158 plastic boxes, which is more than enough to fill, you know, every single shelf with the plastic container. So we're pretty good for expansion space for the collection. And the cultures, now that we have that new room and those new cabinets, and the department is very supportive of what we're doing now. So there's definitely, if we needed it, they would be here to support us. We have very good management all the way up to Scott Hansen, who runs DPI. He loves what we're doing, loves all our outreach engagement, loves to do stuff like this, promoting DPI, promoting the collections. So no problem there. We have a small library in the herbarium with actually a lot of sort of historical books and heritage books. And then we have the actual library here, which it's very dated. It's like up in the back shed now, and I don't know, a lot of things have been digitized. We're consolidating. I think a lot of our department libraries have been shut down and moved here. But that's just, you know, the landscape is changing. People don't need books as much anymore. And certainly, I don't know, people have their own personal references to many of them, or they rely a lot more heavily on offline resources. But we try to, you know, keep a collection in the herbarium, especially with mycological related texts. And there's certainly some rare things in there that you can't get a hold of. So, yeah.
Interesting. All right. Well, Susie, did you want to ask your last question and
I will
wrap up?
Okay. Jordan, when I visited the herbarium, or my fungarium in New Zealand, in Auckland, they have a room that is not considered to be New Zealand soil. It's like an embassy is not. So the embassy of Russia on Australian soil is not Australian soil, it's Russian soil. Do you have one of these rooms? Did we see that earlier?
No, but is that how they get away with importing cultures from overseas? Because, like, literally, we have, so one of our trustees, Mr. Burgess, he was one of the foundation members for the Australian Mycology Society. He's highly esteemed in plant pathology. He does all this outreach engagement overseas with Laos and Vietnam and collects all these cultures. Very important for us to know about emerging pathogens, what's a threat, what's coming, and he can't bring them into this country. Yeah, because you have to have them ID to species and apply for a permit for every single species. It's like $450 just to apply with the guarantee of approval, and it takes weeks. So he sends all these cultures to New Zealand.
Wow, okay.
He sends them to his colleague in Italy as well. So Yeah, it's on our agenda with, I'm part of the National Plant Biosecurity Diagnostic Network, and it's one of our things about, yeah, working with government to understand that we are the biosecurity collection, like we're importing pathogens to help protect us from them, and we're the best people to make the decision on whether it should be allowed or not. Yeah, so we're trying to work on that, but basically we try not to have, so we do have quarantine level 3 approval for storage. Maintaining approval to work on them was so much effort, we barely ever did it that we just let that lapse. But the Elizabeth MacArthur Agricultural Institute, so the other DPI site in Sydney, has level 3 clearance to work on these sorts of pathogens. But these are things that were important before all these laws were brought in, so they're sort of grandfathered to exotic specimens, and also when we've had an incursion and eradicated it, you know, things still considered level 3 because it's not in Australia, even though it was collected in Australia from when it was here. Yeah. Yeah.
Okay.
But no, I don't know. Foreign soil, fancy. Yeah, I'll have to talk to Evan about that.
Yeah, no, I was quite surprised to think that inside an herbarium here was this little space that was about 3 by 4 meters, and it was not considered New Zealand soil at all. And it was like, oh, I'm out of the country, I'm in the country, I'm out of the country, I'm in the country. It was kind of cool.
They do really cool stuff with liquid nitrogen storage as well. It costs a lot to invest in that and maintain it. I think Bevan said he spends $20,000 a year on liquid nitrogen alone, but that's like the best way to store stuff for that long-term preservation. Because even the things that don't like freeze-drying microbacteria, chuck them in liquid nitrogen. And that's how I worked on my PhD. I was looking at wheat stripe rust, and I was going through the historical collection from the surveys, basically from when stripe rust first occurred in Australia. Every year they have put into liquid nitrogen representatives of what populations are emerging, and you know, because they change their pathogenicity and what cultivars they can infect. So I was able to just pull all of these rust out from liquid nitrogen and then inoculate plants and see what they would do and all of that. So I feel like a retrospective historical study using specimens, which was really cool. So yeah.
Cool. Okay. Thank you, Jordan, for your valuable time and amazing tour. I'm going to have to visit the collections in Orange really soon, I can see, and visit Michael too.
That you know, email me. I'm sure you guys will send my contact. I do speak all the time. More in depth than this, you know, if you want, and yeah, come and see things. We've got other things I didn't get a chance to show you, all our like heritage books and our scanned electron microscope and how we take photos, and yeah, so we're always happy for visitors, and especially if you bring in specimens. So, yeah.
Sounds terrific. Thank you so much. So sadly, we're out of time now. It's been great, Jordan. Thank you. Nice to see you again too, since Roy's workshop back in 2017, I think it was, or 18. For those of you who still have questions, please email them attention to Sophie at fungimapcoord@gmail.com, and we'll endeavour to supply you with Jordan's answers in due course. When you receive your link to the video for tonight, we'll also send a link to Jordan's fundraising merch at Redbubble for you. And if you'd like to join FungiMap, purchase any books, or just have a look, please go to our website, fungimap.org.au. Also, if you have enjoyed our webinar tonight with Dr. Jordan Bailey, you may like to book for our next month's Funginar, which is Steve Young, photographer extraordinaire of all things super, super tiny, namely slime molds. There's a new slime mold book out with some of his fabulous photos in it. I think it's the Secret Life of Slime Molds or something, put out by Cyro. And importantly, how to photograph these tiny things is going to help us with that. More details will come shortly. So keep a watch for our newsletter on Facebook, Instagram, and the website. Thank you, everyone, and good night from Fungi Map. See you at our next Funginar.
Night.
Bye.
Night, Jordan. Night, Sophie. Night, everybody.